Good afternoon, everyone, and welcome to Prescient Therapeutics' special investor briefing on their newly unveiled cell therapy enhancement platform, CellPryme. My name is Patrick Nelson, AMD at Reach, and I'll be hosting and facilitating today's webcast. But more importantly, we're joined by Prescient's MD and CEO, Stephen Yatomi-Clarke, and Senior Vice President of Scientific Affairs, Dr. Rebecca Lynn. Thank you for joining us today. In today's session, Stephen and Rebecca will provide an overview of Prescient's new platform for enhancing cell therapies, called CellPryme, and how it complements their existing next-gen CAR-T platform, OmniCAR. Now, to stay up to date on Prescient's news, type PTX, which is the code they trade on the ASX, into the chat box at any time during the presentation, and we'll make sure that you're kept abreast of investor updates. We expect the session to run for approximately 30 minutes, and there will be a Q&A session at the very end. If you'd like to ask a question at any time, simply type it into the question box, and we'll put it to Stephen and Rebecca. We'll just see how the session's flowing, but we'll deal with all sessions at the end if we don't get to them as we go. And we really like the sessions to be interactive, so please put forward your questions while you've got the guys. It's a good opportunity. Now, any advice contained in today's presentation is general only. It doesn't take into consideration your personal circumstances. Past performance is not a reliable indicator of future performance, and the information we're giving you is for educational purposes. Now, just a quick introduction onto PTX. So today's session is about CellPryme, and if you would like to get a more detailed overview of PTX, we'll be hosting a investor briefing to go through what they do in entirety in a couple of weeks' time. So just type yes into the chat box if you would like to be invited to that session, and we'll coordinate an invite. PTX is a biotech company predominantly active in CAR-T, which is a cell therapy promising to revolutionise cancer treatment. The company owns a number of highly attractive assets that have been developed with world-leading partners, and these assets can be found grouped into platform technologies and individual therapies. They're two platform technologies creating next-gen CAR-T therapies. The first is OmniCAR, which is a modular cell therapy developed by University of Pennsylvania and Oxford, and has the potential to make CAR-T more accessible, cheaper, safer, more effective. The second platform, which we're going to be talking about today, is CellPryme, which is a cell therapy enhancement developed by Peter Mac that enhances current CAR-T therapies, or current generation CAR-T therapies. Both platform technologies enable PTX to partner with any therapy provider, thereby they're effectively, it's sort of a shovels to the gold rush positioning, making their business scalable, diversifying the risk, steep upside, earlier and more certain revenue. In addition, they have 3 in-house therapies based on OmniCAR and developed with Peter Mac, targeting highly underserved cancers in AML, GBM and solid tumours. And they also have 2 clinical stage targeted therapies addressing mutations inside cancer cells, and they are PTX 100 and PTX 200. And despite the platform's highly attractive model, the therapies are essential to PTX. Each of the 5 therapies in itself provides significant commercial upside, with PTX 100 and 200 well advanced into clinical phase 1B. The next 3 generation therapies provide proof of concept for both platforms. All of PTX assets are a premium pedigree, having been developed by and with Yale, Moffitt, University of Pennsylvania, Oxford, Peter MacCallum, and other leading industry partners. With ongoing significant news flow, the company is advancing towards becoming the central platform of the CAR-T revolution, right? But today we're here to hear about the recently announced CellPryme platform, which produces better and more effective cell types for current and next generation CAR-T cell therapy. CellPryme is easily incorporated with existing CAR-T manufacturing processes. Simply put, CellPryme-M platform produces superior cells during the cell manufacturing process, which are basically fitter, less prone to exhaustion, enable longer duration of cancer-killing activity, developed by Prescient in collaboration with Peter MacCallum Cancer Centre, obviously a world-leading cancer research institute, but PTX retain the whole ownership of the intellectual property, and it can be used for their own therapies, but it's also can be applied to any CAR T therapy, which opens up the opportunity to literally hundreds of partnerships. Very exciting development for PTX. So today we've got CEO Stephen Yatomi-Clarke and Senior VP of Scientific Affairs, Dr. Rebecca Lin, who will be running through the CellPryme platform in more detail. Stephen, I'll hand the controls over to you.
Thank you very much. Thanks, Pat, for the great intro and synopsis, and many thanks, everyone, for taking time out of your day to join us. Yeah, we're absolutely delighted to unveil CellPryme. For a long time in our pipeline slide, you would have seen cell therapy enhancements undisclosed for a little while, so this is what we've been developing in stealth mode with Peter Mac. Why was it in stealth mode? Well, for commercial and intellectual property reasons. We just finishing up the data, finishing up the experiments and submitting the patent so we can finally go public with it so that we can protect it for the value of all shareholders. So we're unveiling CellPryme in two parts, and CellPryme-M is the first one. So this is all about enhancing cell therapies. So just to take you through what that means, clicking through. So yeah, as Pat says, this is another platform which is wonderful to enhance cell therapies. This is relevant to current generation cell therapies, things that people are developing now and is also already in the market, but also a next gen. And as such, it is really complementary to OmniCAR, and I'll explain how they fit with one another. Basically, CellPryme-M produces a better cell type, a better, more youthful cell type. The upshot of it is it doubles tumour control and survival over regular CAR Ts. It really turbocharges those cells to do a much, much better job. It does this by making them last a lot longer so that they can hang around and kill the cancer for much longer, and also by helping them actually traffic to the tumour site, locate the tumour and penetrate the tumour mass once it's there, which is especially important in solid tumours. There are many development opportunities in front of CellPryme-M, internal and external. So internal, yes, we'll be using them with OmniCAR to turbocharge those programs and really take that to the next level, which is incredibly exciting, but it opens up a lot of external collaboration opportunities as well. And importantly, this is something that we can drop in with minimal intervention into third-party manufacturing processes so that they can produce better cells as well. And we'll explain how that sort of is a minimal interventional step later on in this talk. CellPryme-M is clinic ready, so that is we've been busy working with Peter Mac in the background, and the result is that we've got enough robust preclinical data now that this is now ready for clinical testing. So definitely hitting the ground running with this program. And as Pat says as well, even though we developed this in collaboration with Peter Mac, Prescient retains all the intellectual property, which is a very strong position. This means we haven't licensed CellPryme-M, which means we get to keep all of the pie, which is nice. To give you an overview of what CellPryme-M is, so what does it do? It's a single rapid intervention into a manufacturing step, and the result is it produces better cells that kill cancer better, the bottom line is. So these cells last longer. If you think of cells, and Rebecca will speak about this later on, it's not a single cell type, it is almost like a soup. And it has certain types of cells in it. What we do is produce a better composition, a better cell type. So there's more memory cells, as the name suggests. These cells hang around a lot longer. It doubles the proportion of helper cells, and again the name gives away the role of these cells. They synergize with the effector cells to kill cancer and help them last longer and kill tumours for a lot longer. And as a result, this doubles tumour control over conventional CAR T. But also, these are cells that can locate the tumour better. They have more chemokine receptors. Think of these as like a a nose to sniff out the cancer and locate the cancer to find where it is and get to where it's needed. And once it gets to where it's needed, it can penetrate those tumours. This is not so important in blood cancers where the tumours are circulating in the same veins as where the CAR T cells are circulating. But in solid tumours, this is a big problem. You need to actually seek out, you've got to exit sort of the circulation and find where these tumours are, and these cells are able to do that. So how does CellPryme work? So again, it's a rapid interventional step. It's just a 24-hour window in what's a standard manufacturing protocol. And what it does, we used our knowledge of biochemical pathways to work out how to intervene and push these cells towards a more youthful, favourable phenotype. And it does this by rapidly down-regulating a certain group of genes and up-regulating another group of genes. And between these, this pushes these cells from a more mature and easily exhausted phenotype to one that's a lot more youthful. So just to give you an appreciation of how it fits in with OmniCAR, as a reminder, the magic of OmniCAR is in its modularity. So we seek to separate the actual immune cell from the binding component, and that confers all of these amazing advantages, multi-targeting in a true plug-and-play system to be able to redirect the cell from one cancer antigen to another to give the clinicians control to enable a more safe administration of a very vigorous therapy. And importantly, it's agnostic on the target and the cell. So we can target any type of tumor antigen, and we can use any type of cell. So OmniCAR is not wedded to any cell type, and that's how CellPryme complements it, because what CellPryme does is produce a better type of cell that lasts longer and can find the tumor. But just to lay this out so there's no misunderstanding, whilst this can work with next generation cell therapies, guess what? It can work with current generation cell therapies too. And we believe this has utility, needs to be explored in other cell types as well. CellPryme-M can work with T cells. It might work just as well with NK cells and other cell types as well. So on that note, I will hand to Rebecca. Many thanks to Rebecca while she's getting control for really overseeing all of this amazing research at Peter Mac and working very hard with the team there. And also would like to thank Phil Darcy and Jasmine Lee and all of the team there at Peter Mac, who are just wonderful to work with and are thrilled with these results. So that'll be me for now, and I'll look forward to joining you later on in this presentation.
Thanks, Steve. And thank you again, everyone, for joining us on this call as we unveil CellPryme-M. So just going into a little bit of a slightly more technical detail about CellPryme-M, the problems that CellPryme-M aims to overcome. So as Steve has already mentioned, CAR T cells, it's easy for us to forget that the CAR T cell products are actually quite complex, and they're not just a single cell type. Because we give them a single name doesn't mean that you've got one homogeneous cell product. Essentially, every CAR T product that goes out is actually a mixture of cells. T cells themselves are extremely heterogeneous, and each one of them has their own unique fingerprint. And this little figure over here just shows CD8 positive cytotoxic T cells, and just that group of T cells alone actually has quite a complex mixture of cells. And this little fingerprint, essentially the genes that they express, dictate where they go and what they do. In the interest of time, I won't go into too much of a T cell biology lecture, but to give you a sense of the two main cell types that we're interested in when we're making a CAR T product, first we have the more commonly referred to cytotoxic T cells. These are our CD8 T cells. When they bind onto a tumor antigen via the CAR construct, They then release their cytotoxic payload. This is where cytokines like interferon gamma and TNF-alpha are released. You get granzymes and perforins, and that's what bursts the tumor cell. That's what kills the cancer cell. However, they don't act on their own because you've got your CD4 positive helper T cells, and when these guys bind onto the tumor cell, they then release cytokines and growth factors that are important for sustaining the CD8, the cytotoxic T cells. So these are two very important cell types that are in the CAR T product that are required. You need both of them, not just one of them. Both of these cell types are what we're aiming to modify using CellPryme. When you look at memory T cells, that's something that's required for clinical efficacy because that is essentially what, the memory, the persistence of those cells is what gives you the longevity of the therapeutic effect. And what we're challenging and the paradigm shift that we are offering here with CellPryme-M is the ability to alter this without changing the length of the manufacturing time. So to appreciate this, I'm just going to take you through how T cells mature. So on one end of the spectrum, we have naive T cells. So these are T cells that have never seen an antigen, tumor or otherwise, before. They turn into stem memory T cells as they mature, then central memory T cells, then effector memory T cells, and finally to effector T cells. Now, both ends of this spectrum, so T cells don't switch from one end of the spectrum to another. It goes through this sequence of steps to mature. And on one end of this spectrum with the effector T cells, these are the ones that are extremely cytotoxic, so they can kill tumor cells really quickly. However, they are prone to exhaustion and cell death. And on the other end, while they are capable of self-renewal and persistence, naive T cells don't have the ability to recognize tumor cells. So while CAR T cells, the common CAR T manufacturing at the moment produces CAR T cells that are along this side of the spectrum, what we're proposing is that we want to make more of these types of cells, the stem memory and the central memory T cells. And what we're offering with CellPryme-M is a shift towards the central memory T cell type. What's important here as well to appreciate is that while these two cell types, the stem memory T cells and the central memory T cells, are capable of self-renewal, they also express much higher levels of CXCR3. Now, this is a chemokine receptor that is responsible for the T cell getting attracted to the tumor. So in addition to chemoattraction to the tumor antigen, you also have chemoattraction towards the tumor simply by CXCR3 signaling. And what we want to be able to do is get more of these cells that can self-renew and take themselves closer to the tumor. Very important for solid tumors. So as Steve has already mentioned, CellPryme-M works well with OmniCAR, but OmniCAR already addresses quite a number of, or nearly all of the hurdles that are commonly seen when it comes to developing CAR T therapies. However, with CellPryme-M, what we're hoping to do is actually offer some of these solutions as well to whether or not you're developing a conventional CAR T cell or next generation CAR T cells. So addressing the concept of the hurdles of poor expansion, and by not increasing manufacturing time and cost and possibly even reducing manufacturing time and cost. We will also be improving trafficking, tumor penetrance, as well as exhaustion and persistence of the CAR T cells. But the proof of the pudding is in the eating, so let's look at some of these characteristics and attributes and then a little bit of data later on. So we want to have better persistence, and that is achieved by preferentially expanding the central memory T cells. And to give you a sense of how long memory T cells can persist, it's been known quite commonly that they'll persist for decades, but we have also seen publications showing that central memory T cells can exist for as long as 75 years. We want to, by improving CXCR3 expression, that chemokine trafficking, to get us better tumor penetrance and better trafficking, very important for solid tumors. We've also found that with CellPryme-M that within as short as 15 minutes, we see increased genomic stability. So the genes that are associated with DNA repair that's important for genomic stability, those are upregulated quite significantly. That's within 15 minutes. We also find that antiviral effects of these T cells are significantly upregulated as well. And this is perhaps a subject for a different briefing, but this is something that we do want to investigate and certainly is a characteristic of cells manufactured using CellPryme-M. Looking now into the data, what we found was that we get 50% more central memory T cells compared to conventional CAR T manufacturing methods. So going from left to right, CellPryme-M over here, these are the cytotoxic CAR T cells. And when we manufactured them, this is from the same donor, when we manufactured them using conventional methods, we get central memory T cells for sure. That definitely exists. However, when you treat them with CellPryme-M or using the CellPryme-M method, same manufacturing timeline, you get much higher numbers of those central memory T cells. And when we quantified this across different donors, so these are different human donors where we've made CAR T cells with and without CellPryme-M, again, same manufacturing timeline. You can see in these open white bars over here that the effector memory T cells, that's actually reduced. So you're reducing the cell type that's more prone to exhaustion. And conversely, when treated with CellPryme-M, when using the CellPryme-M manufacturing method, you get this converse effect where you get increased numbers of central memory T cells compared to conventional methods. And when you administer these cells into animals with a breast tumor, a very aggressive breast tumor, you get a much better tumor control and increased survival. So let me take you through this data. So in this case, these were syngeneic HER2 C57 black 6 mice. So that's a lot of acronyms basically to say that these are animals that already express HER2. And they have an intact immune system. In these animals, what we did was we then transplanted breast tumor cells that grow very aggressively. We transplanted them into the mammary fat pad. So we're putting in the breast tumor cells where breast tumors normally would grow in mice. And these are mice with a full immune system. So if you're going to get adverse events in these animals, you will certainly see them because it's not for the lack of an immune system, not lack of rejection. So in the black line over here, what you see are the control animals. So these are the animals that were given a tumor, and you can see that the tumor grows very aggressively, very quickly. Within 14 days, the animals then had to be humanely killed for reasons of animal ethics. So these animals the tumor grew too large and the animals were too unwell for us to keep them on the study. So this is why that line terminates over there. When we give them a conventional CAR T, so CAR T cells made using conventional methods, you get tumor control. But this is not significant. So that's what this N.S. stands for. It's not statistically significant. And in fact, you don't see much separation until around about day 6 on the study. And then there is some indication of tumor control later on. However, if you give these animals CAR T cells manufactured using CellPryme-M, you can see even from the very first time post-inoculation that we start measuring the tumor differences, you can see that there is already a size difference and some tumor control early on, and this continues throughout. And this p-value here shows you the statistical significance. So these are significantly different from the control animals. And what you also find is that with the animals that were not given any kind of treatment, they had to be terminated at day 14, and then you get a little bit more control with conventional CAR T cells, but an extension of that survival is actually seen only in the CellPryme CAR T treatment group. And here they are in a bar graph, basically showing the survival of those animals and when we had to terminate the animals depending on which experimental group. And again, the significant difference is really only when you have CAR T cells given CellPryme.
If I might, Beck, if you could just go back just to emphasize one of the points here, and this is a question I got recently, just to be clear, so this is a breast cancer model that is so aggressive that they're normally resistant to CAR T, so the most vigorous type of therapy you can give them. And this is a model that Peter Mack knows very, very well. They know that it's resistant. So we've thrown the hardest tumour at it. We're not being cheeky and giving it to an NSG mice where you're going to get an effect. This is a mouse with an intact immune system with the hardest breast tumour where CAR T doesn't normally work. And what we're doing is we're not changing the CAR T other than with how it's made. So it's the exact same construct on that green line as it is in the blue line. The only difference between the two is how we've made it with CellPryme. So it's the same construct. Another way of explaining that is that we can increase the performance of any one given construct simply by changing how we make it. Would you concur with that, Beck?
Yeah, absolutely. Thanks, Steve. Thanks for clarifying that. And with regards to the persistence and exhaustion, you saw the phenotype that we get, or the type of CAR T cells that we get right after manufacturing. And what we were interested to find out was whether or not we get a continued effect. If that effect that we see, those changes in the CAR T cells, if that is sustained after being introduced into the animals. So what we did was we did a bleed of animals from each one of those groups at day 9 of the experiment. And this is what we found in the blood of those animals. So when we looked at the cytotoxic CD8 T cells and the helper cells, what we found was there was a profoundly increased levels of the central memory T cells in both of those cell types. Conversely, what we found was the effector T cells that we were less interested in, these are the ones that are more prone to exhaustion. They have a cytotoxic effect, but they are more prone to exhaustion. And where you get that loss of persistence, you also get a loss of those cell types. So we get that, so that initial preferential increase in the central memory T cells, that persists even after they've been administered into the animals, which would explain why we get better tumor control in those animals in that very aggressive breast tumor model, as Steve was saying. What we wanted to also ascertain was whether or not the treatment with CellPryme-M actually influenced the potency of cytotoxic T cells, because what you'll remember in that early maturation timeline that I showed you was that on one end you've got very cytotoxic T cells with the effector T cells, but then as you go into the less differentiated, less mature cell type, or the more youthful cell types, as Steve puts it, they tend to be less cytotoxic. So that was something that we wanted to investigate, because we don't want to have that trade-off of having more persistence but less cytotoxicity, because of course cytotoxicity is key to getting rid of the tumor. So what we did here was basically what we refer to as intracellular staining. So using flow cytometry, we measured how much of these cytokines that are used for that cytotoxic payload, that are the cytotoxic payload, how much of it was being expressed in these cell types. So here we've got the control column here. In that top row, these are the CAR T cells that are in the tumor. In the bottom row, these are the CAR T cells that are in the spleen. Why the spleen? Well, it's a secondary lymphoid organ, and it's a nice reservoir for the CAR T cells to kind of hang out until they're needed and then pulled into the tumor yet again. So looking both at the tumor and the spleen, what we found was the conventional CAR T cells, or CAR T cells made using conventional methods over here, you've got 22%. You actually don't get a significant loss of that cytotoxicity, even treating with CellPryme. So that's really encouraging for us, and we found that to be the case, regardless of whether or not you were looking at the tumor or the spleen. And that was with both of those cytokines. So that's great for us. We're retaining the potency of the cytotoxic T cells. We're also not significantly increasing it, because that was the flip side, of course. You could get more cytotoxicity when you alter a T cell phenotype, and that would increase the risk, but we're not seeing any of that. So we're retaining potency, and we don't see increased risk of safety. We don't see increased safety risk associated with cytokine release syndrome. And going back to that CR3, as I mentioned right at the start, this is a chemokine receptor that's expressed on the surface of T cells, and effector T cells can downregulate this. So then that limits their ability to actually track and locate the tumors. However, with CellPryme-M, we find that this actually significantly increases the CXCR3 expression and better trafficking to tumor site. This also gives us better tumor penetrance. And this could be why we're seeing that better tumor control, albeit in a very aggressive breast cancer model. So with that, I'll hand it back over to Steve, who will talk us through steps towards commercialization.
Thank you very much. Thanks, Beck. That was great. Just clicking through. So how do we develop this from here? What do we do with it? So the benefit of this, as I mentioned before, this CellPryme process integrates very easily into standard manufacturing protocols. We're not asking anyone to do anything incredibly differently. And this is, it also bears mentioning that this is not, say, a new drug that needs to touch a patient and therefore needs to go through quite substantial regulatory changes for anyone. This is during the manufacturing process. There'd be alterations to a minor amendment to a manufacturing process, and that should pass through. Anyone looking to make this amendment, it should be quite straightforward. There's no impact on manufacturing time. We know that manufacturing of CAR Ts is very time sensitive. It's currently a little bit cumbersome. The current generation of cell therapies, 22 days vein to vein by the time the blood's extracted, by the time they get the therapy back in, and this doesn't impact on that at all. And even as people are developing much more rapid manufacturing of cell therapies and CAR Ts, this is not a space that we play in, so we're not pretending that we're going to be manufacturers ourselves. What we are saying is whether you're using today's manufacturing process or you're developing a really rapid emerging manufacturing process that we're reading about now, this is a very easily incorporated step. So, and also this has been developed at a GMP grade, which means it is ready for clinical use. So it is clinic ready. It's not sitting there in a less acceptable form and needs to be remade. We've got it ready for clinical use right now. So Prescient will be applying this to our own programs. We'll be our own first customer. That's how much we believe in it. Perhaps not as important for our AML program, but definitely for our solid tumour programs, this will be very, very important. So a way of turbocharging those programs, again, complementing the modularity and all the unique features of OmniCAR by putting SpyCatcher onto the perfect cell is what we're trying to do, and to provide, creating trade secret manufacturing processes. But again, like we have with OmniCAR, which is a platform where we're developing things in-house, but with external opportunities that give rise to monetization opportunities, this is another such platform. Yes, we'll be using it in-house, but there's many external opportunities to incorporate CellPryme-M into third-party programs, and once again can be dropped into their programs with minimal intervention. The lowest hanging fruit, the people who I think are going to be most attracted, we think everyone will be attracted to this using the CAR T space, but for those programs that people are developing that might have stumbled or are a little bit marginal, where an improved cell type can help get them over the line, I think that's the lowest hanging fruit. They are likely to be some of the first conversations we will be having. It is not irrelevant to blood cancers. We believe an increased persistence is a good thing there, and this produces the phenotype that results in better persistence, as Rebecca's described, but especially in solid tumours where, yes, persistence is important, but to get these to traffic to site and do the work that they need to do where they're required. And again, the ability to monetize this platform a lot sooner than would otherwise be the case. So it's a bit to take in, but at the end of the day, it's pretty straightforward with CellPryme, and it's all data backed. So yes, again, it's a platform. It's not a single product with a single application. It can be applied to current gen and next gen. It's complementary to OmniCAR, so a beautiful fit within our pipeline and a great offering. Another 2 platforms sitting there. Produces superior cells, in summary, 50% more memory cells, it doubles the amount of helper cells, significantly more chemokine receptor for better tumour trafficking and penetrance, and the result is it doubles tumour control and survival in the hardest cancer models with immune, with their immune systems intact. Using the same CAR T, it turbocharges the existing CAR T to double their effect. It is ready for the clinic now. We own the IP, and there's multiple development opportunities both in-house and externally. So with that, I will leave it with you. Just as a teaser, we did say CellPryme came in 2 parts as per our pipeline slide. There were 2 programs. CellPryme-A, we're finishing that off also with Peter Mac now, and this will be synergistic and complementary to CellPryme-M. So CellPryme is like a mini family sitting there, all of them relevant to cell therapy programs as they sit today, and future-proofing us into the future. So with that, I guess I'll throw to Pat to host any questions.
Thank you. I'm just going to go back to this slide here, Stephen. A couple of questions around partnerships. And so how many third-party programs can you potentially provide CellPryme to?
Oh, theoretically, there are dozens and dozens. So those are already in. So this is not like OmniCAR, which is really revolutionary in asking people to redesign what they've got in order to give it control features. They're discussions we're having, believe me, and got a very full dance card at an upcoming bio conference next week. But this is a little bit different, where it can be dropped in today with minor amendments to manufacturing. Yes, people will have to stop what they're doing for things in the clinic now in order to make these amendments, but it is a much more accessible universe of opportunities available, and it'd be dozens and dozens of people who this would benefit.
Okay, I'm going to pair up two questions here from Robert and from Ben. So the first part is, how soon can CellPryme bring in revenue via licensing? And the other question was, can you give a sense of when you'd expect first revenue from this IP?
No, you can't. People are very keen to press me on this, when a dollar's coming in, and we're not in the business of making financial projections. Again, we've just unveiled this on Wednesday. Let's just take a cold shower for a second. So we're going to bio next week. We're having our first conversations with people on this next week. It's interesting because we had quite a full dance card before we announced CellPryme, and as soon as it became public, we put out a bunch of supplementary ones, and we got bang, 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 got all these acceptances. So we know people are interested. We know it's relevant. We're doing all the things we need to do. It'd be great to have some people in due diligence on this in the next quarter or two, and we'll take it from there. It'll follow its natural process. So that's about as much as we can responsibly say.
Thanks, Stephen. I asked the next question, and I guess addressing it to OmniCAR and also CellPryme, and from Tony. Realistically, when is the company targeting achievement of its first partnership deal?
That's the third time. That's the third question asked in a different way. So I don't know how often I don't know how many ways I can answer that, but we're doing all the things that we should be doing. Again, I reckon we've had over 300 meetings this year with people. These things take time. If there's a reason you don't see partnership deals done every day with Aussie listed biotechs, it's for a bloody good reason. It takes a long time and it's hard to do. I can think of 2 or 3 examples in the last 10 years. So these things don't, it's not like you put out a flyer and you sign a deal. I don't want to give that impression. These are hard to do, and they typically happen much later in development. What I am saying is that both of these platforms are so unique and so useful that they're attracting attention much sooner than anyone would otherwise reasonably expect. And we are having those conversations. We've got lots of parties under CDA and having lots of parallel discussions, which is exactly, that's the playbook of how it's done.
All right. Matthew's commented and asked, this is very exciting IP. Is PTX doing any work to genetically engineer the starting cells within the soup to introduce or delete specific genes or functions?
There are platform things we're doing with the platform that we can't be specific about, but I'll actually invite Beck to discuss some of the things that we're considering there without giving any IP away, Beck.
That's going to be quite hard. Yeah, I feel like I have been put on the spot. Thanks, Steve. I think the answer is we are reading the trends, and we are doing projections of our own, and we are doing what we feel is best for shareholders in terms of future-proofing our own pipeline. And I think that's about all I can say.
Thanks.
Thank you. I'll ask this question. We'll see what you can say about this one. But what is CellPryme-A that is soon to be released? Please let us know what the outline is and when it might drop. And that was from Jonathan.
You'll know when you know, Jonathan. It's complementary to CellPryme-A. It comes from the same stable. It is looking to It is in the stable of cell therapy enhancements, so that's what it's looking to enhance cell therapies in a different and complementary way to CellPryme-M, such that they can live with one another and synergize with one another.
Gavin has asked, is there has this been peer reviewed? Is there a journal article that he can have a read of?
No, we've just announced it on Wednesday. So the process of submitting it for peer review, that takes, well, Rebecca will be able to opine on how long that takes to submit an article and have it reviewed and all the rest, but we wanted to get this in as soon as possible. But yeah, we have every intention of having it published.
Yeah, so I can comment on the sequence of events if you like. I think that's probably the most reassurance that we can provide at this stage. Our first priority was protecting the IP, which meant that we could not emerge out of stealth mode until all of the patent filings were at a sufficiently mature stage that we could unveil it without getting scooped, as it were. The next thing that we have done is we will be, in addition to slide decks like what you've just seen today, we will be then allowing the Peter Mac team to emerge out of, their embargoes so that they can then present at scientific conferences, academic conferences, where it will receive that level of peer review as we continue to collect the data. So it typically takes, a paper will take anywhere from 2 to 8 years, and I'm not saying that it'll take 8 years, but we're looking to have a manuscript ready probably before the end of the year, and we are, at the moment, we're on track to be collecting data for that manuscript. But even from the time that we submit a manuscript to going through peer review to publication, that is months. So for you to have a paper to read, you're going to get more information out of Steve and I through these sorts of investor briefings than you will from a paper, unfortunately.
Yeah, I think the other thing to say on that is that we're not, that peer review is super important, no doubt about that, but it would make us beholden to like an academic time frame, if you like, without besmirching academia, and we're in a hurry, so we are not waiting for things to be published before we go out and market this. In fact, not only are we speaking about it at Bio, but some of these very science-specific conferences where it's going to be peer-reviewed and presented, even posters and whatnot, that Rebecca will be at herself. That'll be later this year as well. They're already scheduled.
All right. Dean has said, amazing progress, team. Just wondering what the competitive landscape around CellPryme type technology is.
Good question. We've not seen this level of data produced by any group. I know people have been attempting this type of stuff for a while, but this level of data I don't think we've seen. I know that Novartis have a platform called T-Charge, which looks to make their cells, looks to truncate the manufacturing process dramatically. But other than that, there are, I think Rebecca might be able to opine on this a little bit more as well, but not sure of anyone that's producing exactly this.
Yeah, and I think that it's answered in the fact that we've gotten our IP to the stage that it is. So not that our patent's been granted, we're not at the stage where we have a granted patent on these, but the first step that we did when we were looking to submit our provisional patents quite a while ago now was to look at the competitive landscape. So part of getting a patent granted is to demonstrate novelty and inventiveness, and we have sought out the patent attorney's advice around it so we can see that there is freedom to operate and a reason to even protect the IP. So I think rest assured that there is sufficient novelty in here that there is something to protect and something for us to market. The other component of it is, like in the example that Steve mentioned with Novartis's T-Charge, that is quite different to what we're offering in that we're not looking to just offer this for OmniCAR, we are looking to partner. So where quite a number of those companies where you've seen, truncated manufacturing timeframes, you'll notice that they're not looking to offer the methodology to others. They're looking to value add their own pipeline. And I think that's quite a significant point of difference worth mentioning.
David has asked, is offering this manufacturing IP to CAR T competitors giving them a leg up and helping the competition?
I can see why you're asking that. If we're clipping the ticket, then everyone wins. So the objective here is to make better performing therapies. And as the platform owner, that helps us as well, right? So long as, look, would we license this to an absolute direct competitor, let's say who was trying to create an AML CAR T against the same antigens as us and might even be attempting to create some sort of control features? Probably not. That's a conflict of interest. But there is so much out there that there are so many cancers, unfortunately. There's so many antigens. There are so many players trying to tackle things that we're not, that there's a lot of things that we can assist with and clip the ticket on before we need to worry about conflicts of interest. But I hear you. It's a fair question, but I think one to be cognizant of, but not put off by.
If anyone's got any further questions, please put them in. If you would like, and I know a lot of people typed yes earlier to be invited to the next investor briefing, which will be a more holistic overview of Prescient. Type yes into the chat box if you haven't already, and we'll make sure that you're invited. Roy has asked a question / comment, which is on share price, Stephen, so I'll tackle it because it's not appropriate for CEOs to be talking about their own share price. But we did see when the news came out, the CellPryme announcement came out, the share price did rally, and then there was some selling. I think that might have really probably just triggered some people that might have had some taking some profits because PTX has performed so strongly recently. There's no other probably point to make it other than that, and I think there's like plenty of news flow around PTX. I think this is a good example, and I think as investors understand this, they're only going to be more enthusiastic about PTX. All right, let's have a look. Any other questions? Gavin has asked, what does the M or the A stand for in CellPryme?
M's for manufacturing, and you'll find out about A later on.
All right, excellent. All right, well, I'm going to we'll be watching your space eagerly, Stephen. But thank you very much, and Rebecca, also thank you. That was a great presentation and overview of something that is very complex. So I really appreciated that. But Stephen, I'll let you call the session to a halt and have the last word.
Just to thank everyone. I know it's the first time that we're presenting CellPryme, so I'm sure there's going to be some follow-on questions, which we warmly welcome. Appreciate that we're not able to answer everyone's questions on commercially sensitive things, but I'm sure you can appreciate what we're restricted by as well. This is just a wonderful addition to our portfolio. I think shareholders can be really proud of the pipeline we have now. So 2 targeted therapies in the clinic, with showing all the encouraging signs we'd expect to see, both PTX 100 and 200. So that's wonderful. Good foundations to build on. Having OmniCAR was still pinch myself that we've got that platform that's ticking away now when we're head down, backside up, really cranking out that research and development, and it's doing what we're hoping, which is great. And producing 3 programs there, so that it's not just a platform, it's a platform yielding real-world products. And then we've got this that is so complementary, and it's so good when you're winning on the data, because the inherent nature of R&D is so uncertain. And you head into these things doing them for the first time. No one else has done them. So that when the science falls your way and your hypothesis is proven, I can't tell you how thrilling it is to have now two products and 2 platforms yielding multiple products is an incredible thing for the company. So we're in a really great position, and yeah, thank you for joining us on our journey. This is exciting times ahead for Prescient for sure.